Hello and welcome to the WeXtreme Online Academy. My name is Chris and I'll be your tutor for today. In the previous video we looked at questions 16 through 20, and in this video we'll look at questions 21 to 25 of the Cambridge As Biology course code 9700. Question 21, which components could be found in the nucleotides of rRNA? Well, let's just remind ourselves what the bases are of rRNA. We have A, U, C, and G. Okay. Now, just a reminder, let's see, any one of the two different nitrogen bases with a double ring. So if we remember, the ones with a double ring are the purine bases. Okay, and the purine bases are A and G. Okay, so any one of the two different purine bases, okay, we mentioned that. Okay, any of the three different nitrogen bases with a single ring structure, well, this is referring to the pyrimidine bases. Okay, and they are C, T, and U. Okay, now this statement is obviously not true because three bases, well, T is not present in RNA, it's only present in DNA, so there would only be two pyrimidine bases there. And then it says any one of the three pyrimidine bases. Well, so effectively statement 3 and 4 are both incorrect. Okay, because there's only going to be two out of the three possible ones present because it is referring to rRNA and not DNA. Okay. However, statement 1 is very true. It's going to contain any one of the two different nitrogenous bases. So it's going to contain A or G because RNA and DNA can both have these. And any one of the two purine bases, well, again, could be A and G again. Okay. So that leaves you with option B as the only correct option here. Okay. So it's very important that you know what the pyrimidine bases are and the purine bases, what they are, and whether or not it's DNA or RNA by reading the question thoroughly. Okay. Question 22. How many statements are true for semi-conservative replication of DNA in a prokaryotic cell? So if we look at the options here, let's figure out which statements are true. So the process takes place in the cytoplasm. This is very true. Okay. An adenine nucleotide will line up against a uracil on the template strand. Now here's a bit of a tricky one because the DNA is the template strand and uracil will not be present in the DNA strand itself. Now this is something where people might make the mistake and think that's the true statement. So be careful and read the question. Each new DNA molecule will contain one strand from the parent molecule. Yes, that's true. Each new DNA molecule will contain one strand from the parent and then also one copied strand that was created. So statement 3 is correct. Statement 4, if the parent molecule contained 40% guanine nucleotides, each new DNA molecule will contain 20% guanine nucleotides. Well, in this statement, you just don't know. There's a total of 60% other bases in the parental DNA molecule. Now, we just don't know what that 60% is. You cannot assume that you would have exactly the same amount of cytosine as guanine in the original DNA molecule itself. Okay, so statement 4 is incorrect. So only statement 1 and 3 is true, which means a total of 2 statements are correct. So A, C, and D are incorrect. Question 23. Sickle cell anemia is caused by a mutation in an allele of a gene that codes for beta globin polypeptide of hemoglobin. The diagram shows the sequence of bases in the small section of coding strand of DNA for both hemoglobin A, the normal, and hemoglobin sickle cell B globin alleles. Okay, so here's your strands. How will the mutation in the allele result in the production of an altered version of the B globin polypeptide? Okay, let's have a look here at the DNA sequence. As we can see here, this is the area we're talking about that's been affected. Okay, so this is a type of a substitution mutation. Okay, it's only affected one base. As you can see here, A has been swapped to T in this form of mutation. Okay, and in this, it's not affected the overall base sequence. It's not affected the entire amino acid sequence. It's only affected one particular amino acid, which is at this point. Okay, so let's look through the statements and see which ones are true. So it says a tRNA molecule with the anticodon GUG will hydrogen bond to the altered codon of mRNA. Well, if this is our DNA, our mRNA would be C, A, C, okay? And therefore our Anticodon on tRNA would be G, U, G. Okay, and then that would go get the amino acid and put it into the sequence. Okay, so this statement is true at the moment. Let's just look at the other things just to eliminate them definitively. A is the correct answer though, but let's check anyway. All amino acids coded for after the mutation will differ from those in the normal protein. Well, this is not true. As you can see here, it's only affected this one. So GAG, AAG, TCT, they're the same in both of them. Okay, so all these other amino acids have not been altered. Okay, so the mRNA transcribed from the sickle cell allele will contain the codon CAC instead of CTC. Well, no, that's not correct either, because we clearly know that this is the one we'll have in the new DNA. This is what will be made. It'll be GTG. Okay, so option C is incorrect. The ribosome will be unable to continue translation of the sickle cell RNA after the altered codon. Well, this is incorrect, because unless that mutation formed a stop codon, which it has not, then translation will still occur as normal. Okay. Question 24, the diagram shows some tissue types of plant organs. So here you would generally have a root, this would be a stem, and this would be a leaf. Okay, so here we can clearly identify that one here is the phloem. Okay, you should remember that from GCSE. Okay, on the outer region of the stem, this is going to be the phloem as well. Xylem sits on the top of the leaf, and the phloem sits on the underside of the leaf. Okay, so let's have a look and see which one's correct then. So if number one is the phloem, we can eliminate C and D as our options. Number two, well, that's the phloem. Okay, number three is the phloem, and 4 is the xylem. Okay, so this leaves you with option A as the correct answer, eliminating B. Okay, question 25. Which row correctly describes the adaptations of companion cells, phloem sieve tube elements, and xylem vessel elements for their roles? Okay, let's pick something that's really easy that we know already, that's pretty well defined in GCSE and A level. Okay, so let's look at the xylem vessel element. Menoplasma desmata for communication between cells. Well, that's false, because the xylem cells don't communicate with other cells. They're dead, they're hollow, they're lignified. Okay, strong lignified cell walls. Okay, well, that statement's true. So B is a possibility at the moment. Empty cells for water to allow freely. Yep, that's also true at the moment. A thin layer of cytoplasm. No, the xylem does not have any cytoplasm at all. Okay, so that can eliminate option A and D. Okay, so let's focus on B and C here at the moment. Menoplasma desmata for communication between cells. Okay, maybe a thin layer of cytoplasm to resist flow. Well, again, possible. Okay, so let's look at option of the companion cell. Empty cells allowing water to move freely. Well, the companion cell function is not to move water about the place. Okay, so they still contain their cytoplasm and other parts as well. So that is a false statement. Okay, and as a result, you can eliminate option B, leaving option C as the correct answer. So yes, there's definitely many plasmodesmata there for communication between cells in order to be able to tell when it needs to be actively pumped or not, the sucrose into the phloem or not. A thin layer of cytoplasm to reduce the resistance of flow. Well, yes, remember the phloem elements are going to be moving substances in both directions, up and down the plant. Therefore, you need to have a reduced resistance for this as well. Okay. Thank you for watching our video. Please register on the website for a free demo, and I look forward to seeing you in our next video.